Assalamu alaikum dear students. Welcome to the course Principle in Animal Life 2. This is lecture number 38. The topic is genetic variation. This is the second part of the lecture. Discussing agenda for the topic is sources of genetic variation. Lecture outcome. After watching and listening this lecture, students will know about various mechanisms through which genetic variation occurs. Genetic variation is the differences in the genetic constitution of an individual, in the genetic makeup of individuals, in the genetic makeup of populations or species, in the nucleotide sequences of individuals. The genetic variation on which evolution depends originates when mutation, gene duplication, or other processes produce new alleles and new genes. We have already discussed those processes. Mutation is a change in the genetic material of a cell or an organism. Mutation can be chromosomal mutation or gene mutation. Gene mutations are changes within the nucleotide sequence of a gene. Gene duplication is the only medium through which new alleles arises. It is responsible for the evolution of gene families. How? Suppose this is a particular gene, and because of mutation this particular gene duplicated. This particular gene duplicated, and this is the duplicated sequence. Now the original gene will perform its function, and this duplicated sequence is now available to adopt and start functioning. It is a kind of stumpany. It is reserved to be assigned some kind of a function, and usually it will start functioning, and the function which it will eventually start performing will be somewhat related to the original gene from where it was duplicated. So in that situation, this sequence, this duplicated sequence is available for natural selection. Natural selection will act upon it, and if the change was favorable, if it was giving survival advantage to the organism, then this change will be retained in the genome, and it will be propagated to the offsprings and spread into the population. Genetic variants can be produced rapidly in organisms within short generation times. Sexual reproduction can also result in genetic variation. How? Sexual reproduction, where the process of gamete formation takes place. During gamete formation, crossing over takes place, and crossing over is responsible for genetic variability. Thereafter, the two gametes fuse together, which provides yet another opportunity for genetic variability. And thereafter, the random mating also provides opportunity for variability. So new alleles can arise by mutation. Mutation is a change in the nucleotide sequence of an organism's DNA. A change of as little as one base in a gene is called a point mutation, can have a significant impact on organism's phenotype. Mutations are harmful. Majority of the mutations are harmful. They decreases the fitness of the organisms, but very few mutations are useful. They increases the fitness of the organism. We might expect that this would be the case. Organisms reflect many generations of past selection, and hence their phenotypes tend to be suited for life in their environments. So let me give you an example of a useful mutation in the Zoology 1. You must have studied an example of a surface receptor on CD4 cells. This surface receptor provides a site of attachment for the HIV virus, and when this surface receptor is mutated, then it cannot bind the HIV virus. So the person with such a mutation are exposed to HIV virus, but they does not acquire the disease, and the gene which is mutated is CCR5. Another example of favorable mutation is the sickle cell condition in African people. This sickle cell condition obviously occurs because of a mutation, but those people are a kind of immune to plasmodium. They does not acquire malaria because this sickle shaped RBC is not a favorable location for the growth and survival of plasmodium. Plasmodium cannot survive inside a sickle shaped RBC. So the people suffering from that condition are immune to malaria. So here what they want to say is that sometime a mutation occurs which alters the phenotype of an individual slightly, and this slight change at times is favorable in the environment where the organism lives. In some cases, natural selection quickly removes such harmful alleles. means the individuals carrying such harmful alleles and such harmful traits eventually dies, and when an individual with a harmful allele dies, so the harmful allele is also removed from the population in such a way. Indeed, a harmful recessive allele can persist for generation by propagation in heterozygous individuals. So sometimes the allele exists in recessive forms. They are not expressed, but they can be transferred from generation to generation, and a population can have individuals who are carriers for that particular allele in comparison to dominant alleles, which if are harmful, they result in the death of the individual, and as a result these alleles are also removed from the population. So such heterozygote protection maintains a huge pool of alleles that might not be favored under present condition, but that could be beneficial if the environment changes. They are talking about the alleles for which the individual is a carrier, means these alleles, although they are not favorable under the present circumstances, but they are carried by that particular individual or the population, that maybe some time arrives in the future or the environment changes, and these mutant alleles may be beneficial in that changed new environment. While many mutations are harmful, many others are not. Recall that much of the DNA in eukaryotic genome does not encode proteins. So they are talking about the changes in the intronic regions, which don't have any effect on the proteins. Point mutations in these non-coding regions generally result in neutral variations, differences in DNA sequences that do not confer a selective advantage or disadvantage, means the changes in the nucleotide sequences in the non-coding regions does not affect the individual or does not have any beneficial or harmful effect. The redundancy in the genetic code is another source of neutral variation. Even a point mutation in gene that codes a protein will have no effect on the protein's function if the protein composition is not changed. We discussed that in the previous lecture, that sometime even a mutation takes place within a gene, but it doesn't have any effect on the protein formed, and this is because of the redundancy of the genetic code. Remember, synonymous mutations or silent mutations. And even where there is a change in amino acid, it may not affect the protein's shape and function. Sometimes even the amino acid is changed, but this change is not harmful because the amino acid which is changed also has the same chemical properties which so sometime mutation brings about a change in a codon, and the codon starts representing a different amino acid, but this different amino acid is luckily of the same chemical category as that of the original amino acid, so therefore it doesn't have any effect on the protein shape and function. Moreover, as you will see later in this chapter, a mutant allele may on rare occasions actually make it its bearer better suited to the environment, enhancing reproductive success. So sometimes a mutant allele, a mutated allele increases the fitness of an organism. Just in the previous slide we discussed the CCR5 gene which is mutated, then the individual becomes resistant to acquiring HIV. Although it is a mutation, but it increases the fitness of that individual. So finally, note that in multicellular organisms, only mutation in cell lines that produce gametes can be passed to offsprings. So remember that mutations can take place in somatic cells and germ cells, and the mutations that takes place only in germ cells will be transferred to the next generation. In plants and fungi, this is not as limiting as it may sound, since many different cell lines can produce gametes. So, in plants and fungi, the situation is different, where the cells from various portions of the body can develop into gametes. So, this particular statement don't apply to these two groups of organisms. But in most animals, the majority of mutations occur in somatic cells and are not passed to offsprings. Chromosomal change that delete, disrupt, or rearrange many loci are usually harmful. Chromosomal mutations are large-scale mutations, and they cannot be considered as a source of genetic variation, because they are so much harmful that they result in the death of the individual carrying them. However, when such large-scale changes leave genes intact, they may not affect the organism's phenotype. If you remember, we studied that the aneuploidy is harmful, more harmful as compared to aberrant euploidy. What happens in aneuploidy? The gene complement is disturbed. Either some genes increase in number or some genes decrease in number, whereas in aberrant euploidy, a complete set, a complete chromosome set increases in number, so it is less harmful. So here they are referring to aberrant euploidy. In rare cases, chromosomal rearrangements may be beneficial. For example, the translocation of part of one chromosome to a different chromosome could link genes in a way that produces a positive effect. A key potential source of variation in the duplication of genes due to errors in meiosis, such as unequal crossing or slippage during DNA replication, or the activities of transposable elements. Transposable elements are the jumping genes. They are present in our genome, and they are detached from one portion in DNA, and they jump and attach to some other portion, and they are responsible for mutations, because at times they land in the middle of an important gene and induce mutations. However, there are certain RNAs which puts a check on the movement of these transposable elements, and mutations do occur during DNA replication and crossing over, so they are also the sources of variation. Duplication of large chromosome segments like other chromosome aberrations are often harmful, but the duplication of smaller pieces of DNA may not be. Gene duplications that do not have severe effects can persist over generations. Remember, gene duplications are the only source of new alleles. They are the source of gene families. In fact, new alleles can arise through other types of mutations also. They result in an expanded genome with new genes that may take on new functions. I have already discussed here that when a gene is duplicated, the original sequence performs its function, and the new duplicated version adopts into performing some other form of function. So mutation rate tends to be low in plants and animals, averaging about 1 mutation in every 1 lakh genes per generation. So because it's a harmful process, so it's rare, and they are often even lower in prokaryotes, but prokaryotes have many more generations per unit of time, so mutations can quickly generate genetic variations in their population. So the variations are much more faster and common in organisms whose genetic material is simple, and it is even more common in RNA viruses. Why it is more common in RNA viruses? Because in RNA the replication process occurs, another RNA is formed, but there is no proofreading. So if a mistake is committed while replicating an RNA, this mistake is not rectified, and the new virus which is formed has this mistake, and sometimes this mistake enables this virus to develop into a new strain, and this at times poses a problem to develop a vaccine against viruses because they change themselves so quickly. Here what I say in advance, they put it in the next line. So the same is true of viruses. For instance, HIV have a generation time of 2 days. HIV also have an RNA genome which has a much higher mutation rate than DNA because of the lack of RNA repair mechanism in host cells. For this reason, single drug treatments are unlikely to be effective against HIV. HIV is the name of the virus which causes AIDS, human immunodeficiency virus. Mutant forms of the virus that are resistant to a particular drug will tend to proliferate in relatively short order. The most effective AIDS treatment to date have been drug cocktails that combined several medications. So because of this prolific ability of mutation in viruses, one particular drug is unable to get rid of them, therefore a combination of several drugs is used. Sexual reproduction provides another avenue for genetic variation. In asexual reproduction, exact replicas are produced. The advantage of asexual reproduction is that only a single individual has the capability to reproduce, but exact replicas are produced. So all the members of the population has the same genetic makeup, the same genotype, and they are exposed to the environment in the same way. There is no variation whatsoever, whereas the sexually reproducing organisms are at an advantage because there is variation exists courtesy of this process, courtesy of the process of crossing over during gamete formation, the random fertilization. So sexual reproduction also have a predominant role in genetic variation, and those are the three mechanisms in sexual reproduction that are responsible for genetic variation. If you remember the first midterm. So this was the end of the lecture. Thank you very much. Allah Hafiz.